Okay, and everyone on Zoom can see the slideshow? Excellent. Okay, so as I was introduced, my name is Jennifer Smith. I am the project manager on the scientific side for the Rat Genome Database, and I'd like to thank the organizers of this meeting for giving me the opportunity to talk to you today about upgraded genome browsers at the Rat Genome Database that support comparative and translational studies. So most of the people in this room probably don't need an introduction to RGD, but if you do, RGD is a multi-species knowledge base. We hold data for 10 species, including human plus 9 species used as models for human disease. We produce and also integrate standardized annotations for disease, qualitative and quantitative phenotypes, pathway, gene ontology, and gene chemical interactions. And RGD has also developed a suite of tools for finding and analyzing these data. And I'd like to talk to you today about 2 of them. The first one is the JBrowse genome browser. RGD is in the process of upgrading to JBrowse 2. It's currently released on our website as a beta version. It's been implemented for all RGD species, and it shares many of the features with other browsers, such as the ability to search for the name or ID of a genome feature and go directly to that region, the ability to select and view multiple data types in a single view, and easy zooming and navigation across a chromosome. Plus, it provides expanded functionality designed to facilitate comparative studies. And I'm showing you here the link to JBrowse 2, plus I'm giving you a QR code. If you follow that code, it'll take you right to JBrowse 2. So comparing JBrowse 1, which is the genome browser that RGD has been using for probably the last 10 years or more, and JBrowse 2, there are quite a few similarities. For instance, they both would have gene tracks. We import gene models from both NCBI and Ensemble. We have disease-related tracks based on RGD's disease annotations. We have strain-specific variant tracks, plus we have new consolidated variant tracks from the European Variant Archive, or EVA, and all of the data types that are available in RGD's JBrowse 1 browsers are also available in JBrowse 2, plus more. So looking at the new data types that you can find in JBrowse 2, I'm going to talk about 2 of them specifically. We now have epigenetic tracks, including attack-seq peaks and alignments for liver, and peaks for an additional 24 organs, and ChIP-seq peaks for methylation and acetylation marks, and transcription factor binding sites in liver, and denervated and non-denervated gastrocnemius muscle. Plus, JBrowse 2 supports synteny tracks. And to explore the synteny functionality in JBrowse 2, we did, for lack of a better term, a quick and dirty run of Minimap2 to compare the RN 7.2 to a number of the other species in JBrowse, in RGD, and also to all of the other rat assemblies that we have in RGD, and those results are available on JBrowse 2. So this is the view of the new JBrowse 2 showing some of the new data types. The right sidebar, this over here, can display either a track selector, so for turning tracks on and off, or feature details, as it's showing in this case. It's giving information about the MTHFR gene, which is this one right here. And the sidebar is on the right by default, but can be moved to the left of the main genome browser sub-window, since most of us are used to having the track selector on the left side. You can move it over there if you prefer. So what is showing here is a track for genes, one for the rat to human synteny, one for RNA-seq expression BAM alignments, one for attack-seq peaks, no, 2 for attack-seq peaks, 1 for attack-seq alignments, and 1 for the ChIP-seq peaks, and the expression, attack-seq, and ChIP-seq data that's being shown here are all from liver. So it gives a convenient way to compare and view things across the different data types for the same organ. One of the nice things about JBrowse 2 is the ability to stack different types of views within the same browser window. So here I have a linear genome view of rat. And if I click this add button at the top, it gives me a list of possibilities of different views that can be shown. So if I choose, I'd like to add another linear genome view, it gives me this kind of list that shows everything that we have in JBrowse 2 for all of the different species and different different assemblies. And so if I choose the HT38 for human and the mouse 39 assembly, then I'll get a stacked linear genome view that looks something like this, where each of these is separate, but you can look at them all together. And this kind of a stacked linear genome view makes it easy to see that, for instance, in mouse and rat, the ALAS2 gene, which is this one and this one, actually overlap the APEX2 gene, which is this one and that one. Whereas in the human, ALAS2, this one, is actually downstream of APEX2 and doesn't overlap it at all. So the Synteny viewer in JBrowse 2 will give you the ability to view tracks for two different species or two different assemblies in the same genome browser view. Here I'm showing the rat BN 7.2 versus the SHRSP de novo assembly from Dr. Doris's group. And you can see that I'm showing a genes track for the RN72 assembly and a genes track for the SHRSP assembly. And you can see that there are blocks in between that show you the correspondence between the two. And I've selected a section of the genome where there's an inversion between the two assemblies, so you can easily see with this view that they're crossed over. And you can also look at the directionality of the genes and see that they are actually reversed. And I'm showing gene tracks, but any of the tracks that are available for each of these assemblies can be shown in the same view. So you would be able to look at not just genes, but other types of data in the Synteny view. The dot plot viewer gives you a genome-wide view of whole genome alignments between two assemblies. And here again, I'm showing 7.2 versus the SHRSP. And you can see that there's pretty good correspondence between the two. And if I would want to zoom into a region, I can do that and see a little bit more detail. And so you get the idea of the comparison between the correspondence between the two assemblies. The breakpoint split view and circular plot are not currently implemented at RGD, but this is software that is available in the JBrowse. So I'm showing this example from the JBrowse website. And we will be implementing this at some point in the future, but it's a very nice way of being able to look at structural variants. So for instance, here in this example, there are translocations from chromosomes 1 and 2 to chromosome 5. And it's just an easy way of viewing that. So one of the limitations of viewing Synteny in the JBrowse 2 browser is that you can only look at pairwise alignments. You can only see two species or two assemblies at one time. So RGD is in the process of developing or actually redeveloping. Thank you. Redeveloping the VCMap tool, which is a Synteny viewer that allows comparisons across multiple species or assemblies. And so if you choose this on the front page, you go to a configuration page that allows you to choose a backbone species and one or more comparator species. And right now we only have 3. We have rat, mouse, and human, 1 assembly for each in the tool, but we'll be adding more. So then you hit load BC map and it gives you this kind of a view. The overview panel on the left gives a chromosome-wide view of the synthetic blocks. And if you mouse over, we'll let you see if they are reversed compared to each other. The detailed view in the center shows the individual synteny blocks and the gaps in between them. And this is coming from the UCSC chain alignments. So it's the blocks and the gaps from the chain alignments. And when a gene or gene group is selected, the orthology is indicated by a red line that goes from ortholog to ortholog, and information is displayed on the selected data panel with links to the corresponding gene records in RGD. And we also have heat map tracks for variant density in the 3 species. Variants are binned, with the bin sizes dependent on the zoom level. So in this case, the bins are 77,000 base pairs, and the color is determined by the number of variants in the bin, going from blue with no variants to red with the maximum number of variants per bin in that species at this zoom level. And similar heat maps for epigenome data are also coming soon. And with that, I would say thank you. I want to acknowledge the incredible RGD team and all of our funders, our collaborators, the developers of JBrowse and JBrowse 2, and the researchers who contribute data and faithfully use our website and data. Thank you, and I'll take questions.